Good afternoon. My name is Jürgen Richt, and I'm a professor at the College of Veterinary Medicine at Kansas State University. And I will talk today on the susceptibility, transmission, and reinfection of domestic cats with SARS-CoV-2. All the work we do is in a BSL-3 egg facility, which is shown here, the Biosecurity Research Institute at Kansas State. So before I start, I will acknowledge all the people which were involved in these studies. This takes a village to do all this work. In my laboratory, there are many people involved, and I want to mention Bianca, Natasha, Daniel, David, and Jesse mainly for really great work, and the Chang lab at Kansas State who does the pseudoviruses. I want to mention Wensheng Ma, who is at University of Missouri now, Bill Wilson from USDA here in town, and Adolfo Garcia Sastre from Mount Sinai, and the immunohistochemistry and in situ was done at Louisiana State by Mariano Indudeni. And without the funding of internal funds, NBAF, National Bioec Transition Funds, the NIH, with the Centers of Excellence of Influenza Research and Surveillance concept grant, and Department of Homeland Security, this work would not be possible. So I want to today, in the next 40 minutes, discuss the outline. This is the outline of my presentation. Why do we have a cat model for COVID-19? What are the results of a primary infection of these animals? What are the results if we reinfect these animals after 3 weeks with SARS-CoV-2? And then at the end, I want to discuss what are the environmental contamination issues in a cat environment, infected cat environment, and is the SARS-CoV-2 genetically stable in cats? So this is the rationale for why we developed this COVID-19 cat model. We all know that most likely the SARS-CoV-2 was transmitted from bats, either directly or via an intermediate host, for example, the pangolin, into humans, and it's very efficiently transmitted among humans, and it caused the 2020 pandemic. What is known now that not only it goes from animals to humans, but it also goes back. This is a zoonotic transmission. It has a reverse zoonosis. It can be transmitted back to animals, from humans to animals. And for example, here we have lions and tigers, which have been shown to become infected by COVID-19 positive caretakers in the New York Zoo. We have dogs in many countries in the world which have been shown to be infected in COVID-19 households. The same is true with domestic cats. We have a lot of cases where in COVID-19 households, cats got infected and were shown to shed virus. And in one study in Wuhan, they found 15% of the animals which they tested seropositive for SARS-CoV-2 antibodies. And we know from several outbreaks in mink farms in the Netherlands, in the US, in Denmark, that mink are highly susceptible to this virus, and they can show severe clinical signs and even mortality. And there is a report on a mink to human, in 2 cases, a mink to human transmission. However, the detection of SARS-CoV-2 in all these animals I mentioned here is rather limited, considering the extent of the human pandemic. So this is our first study where we did primary infection and reinfection of cats, and I will go through in this diagram how we set up this experimental model. So we have 2 control cats which we necropsied before we did the infection study in cats. We had 6 cats which we challenged per os and intranasal simultaneously with 10 to the 6 TCID50 of the US strain WA1 2020. And we used this where they were separated in 2 cages in 2 groups with 3 cats each. We added, after 1 day, 1 sentinel per cage into 2 sentinels in total after 1 day of infection. And then on day 4, we necropsied 2 infected cats. On day 7, we necropsied another 2 infected cats. On day 21, we necropsied another infected cat. And then we had 3 cats left, which we then reinfected with the same dose of the same virus intranasally and per os. And then we followed these animals for 4 days, and then we did necropsy and looked for pathology, microscopic pathology, and all the virology associated with this study. These are the animal rooms. We keep these animals. These are BSL-3 egg animal rooms, and we keep them in these cages, 4 cats per cage, 3 principal and 1 sentinel animals. And we also do a lot of environmental studies, as shown here with an air sample on the right. And in order to handle cats, we also house them before we sample them and bleed them in single cages, as also seen here on the right. These are our sampling timelines, and we start with infection on day 0, with the sentinels on day 1 added to the principal animals, and we did body temperature and clinical signs daily. We did body weight on every 2nd or 3rd day, and then we also did nasal, oropharyngeal, and rectal swabs every 2 or 3 days. And then on similarly, we did air and environmental samples on various days post-inoculation. We did blood collection also on every 2nd or 3rd days. And we did on day 4, 7, and day 21, we necropsied animals. And we did during after euthanasia, we did nasal washes. We pulled the lung, the entire lung, and did bronchoalveolar lavage fluid both. And we sampled the entire upper and lower respiratory tract, the GI tract, the central nervous system, various nerves, the eyes, the inner ear, major organ, urine, feces, cerebrospinal fluid, various lymph nodes, and bone marrow. In total, we had 10 cats, 2 control cats, 6 principal infected cats, and 2 sentinels. In blood analysis, we did an entire blood analysis at various time points post-infection, and we used the VetScan HM5 to do that. We did white blood cells, lymphocytes, monocytes, neutrophils, eosinophils, basophils, red blood cells. We determined hematocrit, hemoglobin, and platelets, and all these data were in the normal range, except for the platelets, where we saw numbers reduced starting at day 5 post-infection. We did viral RNA detection, PCR, that we used the CDC PCR technology or technique or probes and primers, and we sampled the blood at various time points post-infection. And with our infected animals, we did not find any positive blood, any RNA in the blood of these animals, principal infected animals in red, and the same was true for our sentinel animals. We did not find any RNemia in any of the animals tested in this study. We then did nasal swabs. In nasal swabs, the story is different. The infected animals in red, we see already positive, these are viral RNA copy numbers, genome copy numbers. We see it already in day 1. It's maintained until day 10 post-inoculation and negative on day 14. And with our sentinel animals, we find the first positive ones on day 3, and it's also maintained until day 10 post-inoculation and negative on day 14 and day 21. If we do nasal washes, and we do that at necropsy days, we see on day 4, day 7, high loads of viral RNA in the nasal washes on both animals necropsied, but none on day 21 post-inoculation. And similarly, if we do bronchoalveolar lavage fluid on day 4 and day 7, both animals are positive, and the one on day 21 is negative. We do oropharyngeal swabs too, and here we see, again, like similarly to the nasal swabs on day 1, the principal infected animals are positive on day 1 and maintain positivity until day 10, and are negative on day 14 and 21. And the sentinels are also turning positive on day 3, but are already, and day 5, but are already negative on day 7. Throughout the rest of these, the other 2 weeks. The story is different when we look at the rectal swabs, and these are animals 4.5 to 5 months old. We see very nice shedding already on day 3. In these a little bit later than with the nasal and oropharyngeal on day 3 post-inoculation in the principal animals, and maintained up to day 14, longer than with the other swabs, and then negative on day 21. If we look in our sentinels, we see a similar kinetic, even higher genomic copy numbers than compared with the principal infected animals, but all negative at day 21. We also looked at viral RNA in different tissues. In orange, we see these 2 animals on day 4 post-challenge. In red, the 2 animals on day 7 post-challenge, and day 21, this 1 animal on 21 days post-challenge. And as shown here in the upper respiratory tract, all of these animals are already positive on day 4 and maintained positivity throughout day 21. However, on day 21, the virus RNA load is lower. When we look in the lower respiratory tract, we see a similar results. All animals are positive on day 4 and also more or less on day 7 post-challenge. And some of them, only some of the tissues are maintained positive on day 21, like bronchi and some of the lung lobes, but the majority is negative on day 21. The lymphatic tissue shows that tonsils, tracheobronchial lymph nodes, mesenteric lymph nodes are positive already in day 4 and maintained until day 21, whereas spleen is not positive on day 4, but positive on day 7 and day 21 post-challenge. If we look at other tissues like GI tract, liver, heart, kidney, bone marrow, olfactory bulb, or central cerebrospinal fluid, we see day 4, most cases, in most of these organs, we see positivity, and many of these are already negative at day 21, like in the GI tract, in the liver, in the heart, and in the bone marrow, and the cerebrospinal fluid. Histopathology and IHC, we mainly see on day 4 and day 7, we see inflammation of submucosal glands, as shown here on day 4 and then day 7. We don't see anything obviously in the mock infected animals, very normal submucosal glands, and the same is true on day 21. And when we look on the right side here with cytohybridization, we see very nice on day 4 and day 7, we see positive reactivity in these submucosal gland areas of the bronchi, and if we use immunohistochemistry, it's confirmed in the same area. And everything, and that's only on day 4 and day 7 post-challenge, and the tissue is negative again at day 21 post-inoculation. The animals are able to develop neutralizing antibodies starting at day 10 post-inoculation and still maintain it until day 21 post-challenge. And this summarizes this first part of my presentation, the primary infection of cats with SARS-CoV-2. And this is recently posted in bioarchives, and here is the website for it. We could show that if we infect cats via the intranasal per os route simultaneously with 10 to the 6 TCID50, we do not observe obvious clinical signs. We saw a day 2 temperature spike, but that's the only thing we saw. No real clinical signs. And these are 4.5, 5-month-old animals. We did not see any significant changes in blood cell parameters or biomarkers. The only thing we saw was thrombocytopenia above 5 days post-inoculation. There was no anemia in the blood. The urine was also negative. We saw transient shedding of viral RNA through the oral, nasal, and rectal cavities. And we saw viral RNA regularly in the upper respiratory tract, lymphatic tissues, and olfactory bulb. On all 3 days, we sampled these tissues on day 4, 7, and 21. On day 21, we did not see it anymore in most cases in the lower respiratory tract, in various lung lobes, in the GI tract, in the liver, heart, cerebrospinal fluid, and bone marrow. We saw macroscopic and microscopic lung, trachea, bronchial lesions, and it is a tracheobronchoadenitis. We could not see or did not recognize any interstitial pneumonia. And this tracheobronchoadenitis is associated with the presence of viral RNA and antigen on days 4 and 7, but not on day 21. And the transmission from the principal infected animals to the sentinel was very efficient within 2 days. We still have to finalize our virus titrations and subgenomic RNA analysis, our ELISpots, and our RNA-seq on various tissues we collected at necropsy. We also addressed then the question, are cats which were infected with SARS-CoV-2 susceptible or protected from reinfection with that virus? And we did 2 independent experiments, one on the 4.5, 5-month-old animals and one on 7-month-old cats. And we inoculated these animals 3 weeks after primary infection. This is our first cat reinfection experiment, and we sampled, we did it the following ways. On day 21, which is day 0 post-second challenge, we reinfected 3 cats, which were left over from the initial experiment, with 10 to the 6 TCID50 SARS-CoV-2, again, intranasal and per os. And then we followed them over 4 days, the clinical signs, body weight. We swapped them on day 0, 1, 3, and 4. We did also air sampling, blood collection, and we did necropsy on day 4 and collected the same tissues we collected previously for the primary infection. And these are the results. If we looked at viral RNA detection in nasal swabs, as shown here, the principal infected in red, the sentinels in blue, and in green now we see the reinfected animals. So there was some shedding on day 1, RNA shedding, but the animals were negative on day 3 and 4. And if we looked at nasal washes, on day 4 at necropsy, there was 1 animal which was positive, but the others were negative. If we looked at BALF, all BALF were negative at the time of necropsy, 4 days post-secondary or post-reinfection. We did the same. We looked for RNA in the oropharyngeal swabs and in rectal swabs, and these are the oropharyngeal swabs, infected in red, sentinels in blue, primary infected. And here are the reinfected. We couldn't find anything in the oropharyngeal swabs in the reinfected animals. In the rectal swabs, the situation was a little different. We could find in all animals on day 3 and day 4, day 1, 3, and 4, we could find RNA and was still present at the time of necropsy. We also looked at various tissues, similarly as with our primary infection. This is the upper respiratory tract, and as you see here, in many of these animals sacrificed on day 4 post-reinfection, or 4-DP2C, and many of the tissues of these animals in the upper respiratory tract, we still found RNA after reinfection. If we looked at the same at the lower respiratory tract tissue, as shown here, most of them were negative. Only a couple like bronchi and some lung lobes were still positive. The story is different when we look at lymphatic tissues, similar tonsils, lymph node, spleen. Many of these tissues in these reinfected animals were still positive 4 days after reinfection. And when we did other tissues like the GI tract, liver, etc., we found that still some of these tissues were positive on day 4 post-reinfection, like the heart, like the olfactory bulb, and the bone marrow. In terms of neutralizing antibodies, at the time of re-inoculation, day 21 post-challenge, day 0 DP2C, all of these 3 animals here in brown, we reinfected, had neutralizing titers of about 100, 1 in 100. And these were maintained throughout the 4 days. It doesn't seem there is an anamnestic response. We looked at the histopathological changes in these animals. On the left, trachea with H&E and insider-hybridization, and on the right, bronchi. And as you can see, in the upper part are the mock-infected animals. In the middle are the primary infected animals on day 4 post-challenge. And in the lower row, we have the reinfected animals also on day 4 post-reinfection. And as you can see, on day 4 post-primary infection, you see these adenitis associated with the RNA in the within the submucosal glands in both trachea as well as bronchi. And if we look at the reinfected animals, we don't see any viral antigen present in the respective tissues and no inflammation in the submucosal glands. We did a second reinfection experiment where we used 5 cats, which we reinfected, and we added 1 day after reinfection 2 sentinel cats to see whether the reinfection could lead to transmission of virus to sentinel cats. And we followed these animals for up to 14 days. Again, with daily body temp, clinical signs, body weights. We swabbed them again with nasal or oropharyngeal and rectal swabs. We also took blood at various time points post-reinfection. And then we did necropsy, 2 animals, 2 out of these 5 on day 4, like before. 2 out of these 5 reinfected animals on day 7. And then on day 14, 2 weeks after reinfection, we necropsied 1 of the reinfected animals and the 2 sentinel cats. So what did we see? These studies are not yet, all the data are not in yet, so I would just focus on the data we have. And the most important was nasal swabs. These are in red against, again, the infected and the sentinel animals on day 1, already positive in primary infected, principal infected animals, day 3 in the sentinels. And if we look at the reinfected animals, we see positive positivity already on day 1, and it's also positive on day 4 and already negative on day 5. The sentinels throughout the entire time we could not find any positive nasal swabs. And also none of the nasal swabs beyond day 5 was positive, beyond day 4 was positive for RNA in the reinfected animals. If we look at nasal swabs, on day 4, both animals which we necropsied were negative. However, we saw really in both animals at day 7, we see positivity. And all the 3 animals were negative on day 14, including the sentinels. Similarly, if we look at bronchoalveolar lavage fluid, they were negative in the reinfected animals on day 4, 7, and 14, and also the 2 sentinels were negative on day 14. We looked at virus neutralizing antibodies, and here on day 21, post-primary challenge, the 5 animals we reinfected are shown with the brown background. They all had antibody titers, neutralizing antibody titers against the SARS-CoV-2, and our sentinel animals were negative. And when we looked at the time of necropsy, they had still antibody titer day 4, the 2 animals, at day 7, the 2 animals we necropsied, and at day 14, post-secondary infection. And none of our sentinels, however, seroconverted. Interestingly, on day 7 and day 14, the antibody titers compared to the reinfection titer was higher, so there seemed to be an anamnestic response. But it's very clear that our sentinel animals, which were in the room, did not seroconvert. So apparently the virus shedding these reinfected animals did not lead to infection and seroconversion of these sentinel cats. So this summarizes the second part of my talk. After reinfection of primary infected cats, we could not see obvious clinical signs and no viremia, no change in blood cell parameters. We had limited RNA shedding, especially via the upper respiratory tract in the nasal cavities, but also in the oral cavities, especially in the second experiment I didn't have time to show, and in the GI tract. We had limited RNA in nasal washes and in the BALF and in the lower respiratory tract tissues. And we had significant RNA still after reinfection in the upper respiratory tissues, lymphatic tissues, and various organs. However, at postmortem, and we only have done it in the first reinfection experiment on day 4, post-secondary infection, we could not see any enhancement of respiratory disease and no pathology, and also no antigen or RNA in lung tissues. We saw an anamnestic immune response after reinfection, especially in the second experiment where we had day 7 and day 14 post-secondary infection. We did not see it an earlier day on day 4. However, and most importantly, the shedding of RNA by these reinfected animals were not able to induce disease, viral RNA shedding, significant RNA shedding, or infection of the sentinels, because they stayed seronegative even 2 weeks after co-mingling with the reinfected cats. So we conclude from these that SARS-CoV-2 infection in cats induces at least humoral immune responses, maybe also cell-mediated immune responses, which we have to still test, which provided protection against re-challenge. And so this means that immunological approaches to prevention of SARS-CoV-2 and treatment of SARS-CoV-2 may be possible. Okay, this brings me to the last part of my presentation: environmental contamination and SARS-CoV-2 genome stability. So we looked, because this is an important question for public and veterinary health, what are the hotspots in an area where cats, SARS-CoV-2 infected cats, are housed? So we took swabs from various areas within the cage, on the cage bars, water bowls, food bowls, in the litter box, cat toys, our boots outside. We did air sampling, we did floor samples, treatment table, the cat fur, etc. And viral RNA load is shown from green low to red high. And this is a summary here. Hotspots we found are the food bowls, the treatment table. We find still very high loads in cage bars, in the bed where the animals sleep, in the water bowl, and significant viral load, RNA load on cat fur, on the shelf, on the cat toys, and less viral load in boots, room air, and in the litter. So an important question for us was to determine whether SARS-CoV-2 infection in cats can generate adaptive mutations. So to remind you how these studies were done, we used the strain USA WA1 2020, passaged it 3 times in Vero E6 cells, and then we transmitted or infected cats. In this case, these studies are the 4.5, 5-month-old cats by intranasal pulse inoculation. And then we asked the question, do they maintain, is the consensus sequence, which is known for WA1 2020, maintaining cats? And how did we do that? We did next generation sequencing using a tiled amplification approach, next generation sequencing approach called Arctic version 3, and we sequenced these by an Illumina NextSeq instrument. So these are the data. We did find very important, we found 2 mutations, amino acid substitutions in the ORF1A and 1B. But more importantly, we found within the S, the spike protein, an insertion. And this insertion is a 12 nucleotide insertion in the NTD, the N-terminal domain of the spike protein, and the amino acid sequence inserted is KLRS. And this insertion was found in all 6 primary infected cats. So we found an insertion in the spike on amino acid position 215, a KLRS insertion. When we looked at the structure of the spike protein and where this insertion is located, it is shown here. On the left, you see the spike, the trimer of the spike protein. In green, you see the receptor binding domain, and underneath in blue, the N-terminal domain. We did then structural modeling to predict how this insertion will look like. And as you see now on the right, the wild type is on the left side, and the mutant virus or the mutant NTD is on the right side. You see that these KLRS, these basic residues, they determine or they build a surface exposed loop here shown in red. So structural modeling predicts that this leads to a surface exposed loop within the N-terminal domain of the spike protein. We then wanted to determine how the mutant virus, the percentage of the mutant virus changes within a defined sample. From the stock virus via the various passages in Vero cells, and we use P3 to inoculate it in cats, in various cell cultures from swine and so on, and into hamsters. And here are on the left now shown the cell culture data. So this insertion goes up to passage 3. We have, it's still not the consensus sequence, but about 25–30% of the P3 passage virus in vero cells contains this KLRS sequence. And when we infect porcine kidney cells, porcine swine testicle cells, this mutation or the mutant virus almost disappears. Similarly, in human liver cells, HUH7, however, it's maintained its percentage in human kidney cells. If we look in cats, and this is the inoculum, and we look at day 1 and then at day 3, 5, etc., and different tissues, we find that the percentage of these mutant increases and becomes the consensus sequence after day 3 post-inoculation in cats. And this is shown throughout various tissues, GI tissue, rectal swabs, various, and even in here in respiratory tissue. If we do the same with hamsters, you can see that we have an increase also in this mutant sequence if we transmit that virus, that P3 vero stock virus into hamsters. The most important or very important question is now, how will neutralizing antibody recognize that mutant versus the wild type? And in order to do that, we developed together with Dr. Cheng at Kansas State lentivirus-based pseudoviruses, which either express the wild type spike or the mutant spike protein. And then we tested them either in a GFP or luciferase-based readout system. And for this test, we use cat sera, various cat sera, a rapid serum, and 2 human sera. And on the right here, you see the wild type titers, and in the middle, the wild type titers, and on the right, in brown, with a brown background, you see the mutant virus titers. And as you can see, for the cat, for the rabbit, and for the human sera, there are not much differences. Maybe a little bit for the human sera, where if you hide a higher titer with the mutant versus the wild type virus, but it seems to be there are no differences in neutralization titers between using the mutant virus versus the wild type pseudovirus. We also developed right away a diagnostic test that's shown here, and we have a common forward and reverse primer, and then we have a wild type probe and the mutant probe. And as you see here in a mixed population, it recognizes wild type in blue and mutant virus in red in a mixed population. And if we have blood purified wild type, it only recognizes the wild type virus. And if we have blood purified mutant virus, it only recognizes the mutant virus. So these RT-qPCR diagnostic test is able to differentiate specifically and highly specifically and sensitive between the mutant with the insertion and the wild type virus. So this brings me to the end of this part. We have shown that the environmental contamination by SARS-CoV-2 infected cats is mainly in the food and water bowls, the treatment table, cage bars, and the bed. And we have shown that the virus, which is transmitted from into cats, that the virus, which is transmitted, amplified for 3 passages in vero cells, selects a mutant virus, which has a 4 amino acid insertion, a KLRS insertion, and we found that in all cats after 3 days post-inoculation. And this insertion is present also in the virus stock material, which we have shown, but in less percentage. It's not the consensus sequence. However, it's the consensus sequence in the cat. And structural modeling predicts that this insertion consists of basic residues in a surface exposed loop. Neutralization assays done so far with pseudoviruses and using positive human, cat, and rabbit sera do not show differences in titer between using a wild type or mutant virus for the virus neutralization tests. And in order to do future epidemiological studies, we have developed a diagnostic assay for wild type and mutant virus, which can be done in a single tube reaction. Again, this can only be done with many collaborations, and I have mentioned my collaborators at Kansas State, University of Missouri, USDA, Mount Sinai, and Louisiana State already, and it cannot be done without the funding provided by the different agencies listed here. Thank you very much for your attention.